Okay, in this video, number 2 for chapter 3, we're going to look at the cytoplasm, the cytosol, and the organelles. On our PowerPoints, this corresponds roughly to slides 14 through 25, and again, that is for the new 16th edition of the Holes, Human Anatomy and Physiology. But again, this information is kind of standard across the board. But so we talked about in the previous video a composite cell and how composite cells generally are going to have everything put onto one cell so we can study it. But when we look at the general cell, we said that there's 3 parts: nucleus, cytoplasm, and then the plasma membrane. So the cytoplasm, I wanted to make sure we made a point of saying that the cytoplasm is not just the liquid of the cell, which is a lot of times what people have been taught. It is the organelles that are in there, the network of membranes that we'll call the cytoskeleton, and the liquid, which is officially called cytosol. All right, so the cytoplasm is cytosol, the liquid, plus the organelles that are suspended in it. Now again, the cytoskeleton, which is just the framework, we'll kind of briefly look at them. It's just the protein rods and tubules that form the scaffolding inside the cell that allow for the structure, but also for things to move around as well. Now, going through these organelles, first one we have on our list is called ribosome. Most people know ribosomes by this. A lot of these will know by one little phrase, and protein synthesis. Ribosomes are the site of protein synthesis. This is where our proteins are made. Now, at this point, we haven't talked about DNA and the process of transcription and translation, which is how I copy down the genetic code and then take it to the ribosome and translate it into a protein. But we'll see that a little later on in chapter 4. And again, it is important to remember that that is where this happens. So ribosomes are the site of protein synthesis. Now, there is a tubing network called the endoplasmic reticulum, and it comes in basically 2 forms: rough ER and smooth ER. Rough ER is called rough ER because it has ribosomes all studded in it. So if it has ribosomes in it, and ribosomes are the place of protein synthesis, that goes to stand to reason that the rough endoplasmic reticulum, because of those ribosomes, is also where protein is going to be made. Now, smooth endoplasmic reticulum is going to be mainly for lipid synthesis. And one way to remember this is lipids are generally smooth. If you've had olive oil get on your hand, or even butter, it's slippery and it's smooth. So one way to remember lipid synthesis is in the smooth ER is by that. Now this is a picture of the two. You can see this is an actual picture with an electron microscope to show, and you can see all these little dots. That's why they call it rough endoplasmic reticulum. Now the layers are still kind of like the same as our very similar to the cell membrane. It's a phospholipid bilayer. And so on the rough ER, we've got ribosomes on it, and in the smooth ER, we don't. Now, the next thing is, and I'm going to jump to this one in the middle, the Golgi apparatus. So these are always going to be associated really with the endoplasmic reticulum, especially the rough endoplasmic reticulum, because what this is basically the post office or FedEx, whatever you want to use. It is a area where I'm going to receive the proteins that were made in particular. But it's more than just that. But we get the proteins that were just made in the ribosomes. It is going to refine it, make sure it's special tertiary structure. If you remember, as we looked through that last chapter, the tertiary structure, how it looks and is bent, is correct. And then it's going to package it, and it's going to address it and send it on its way. All right, so the Golgi apparatus is basically like a post office. And the things that it packages, the packages it makes, are called vesicles. All right, all a vesicle is a membranous sac. It's used to store and transport substances, more than just the Golgi apparatus. I mean, we'll see them in different areas. We'll see how the cell membrane and allowing things to come or go might make a vesicle. But all a vesicle is a membranous sac. And the easiest way to see it is in the Golgi apparatus because that's what it makes. Now this little bottom part trying to show a interaction of the organelles, I'm not going to go through this. This is not something I want to test you on, but it's just talking about milk secretion and how it's an example of the different organelles working together. But I'm not going to, that's not part of this test that we're going to have. So we're just going to concentrate on the organelles. So this picture right here is showing the Golgi apparatus here on this purple. This is the way it looks, again, electron microscope. So we're making proteins on these little areas of ribosomes. It's going to be transported into the Golgi apparatus, and as it moves through, I'm going to pinch off certain parts of its again, phospholipid bilayer and make little packages that we call vesicles. And then it goes to wherever it needs to go. The next one is a pretty famous one. Most people know this as the powerhouse of the cell, right? So the mitochondria is called the powerhouse of the cell. And we're going to see that this is the site of aerobic respiration. And again, next chapter, we're going to look at ATP, which is the molecular energy horse of our body. It is what drives our body. And the mitochondria is where we make ATP in abundance, but it needs oxygen to get to it. And we'll see that when we look at cellular respiration. One of the things I'm going to look at in the picture of it is showing you how it has this special membrane organization inside of it. And again, we'll study this more in cellular respiration. These next two are specialized, basically specialized vesicles, lysosomes and perioxiomes. So lysosomes, first of all, lyse means to break down. It means to cut in half or break down. So a lysosome is a special vesicle that it contains digestive proteins. And these digestive proteins in this vesicle, what will happen is if I have some sort of organelle that isn't, it's older and needs to be removed, or any waste products like proteins or carbohydrates, this will engulf it and dispose it. Anything that can be recycled, it will recycle, but things that it can't, it just breaks down. And that's why they're referred to as the garbage disposals of the cell. The next one, which are called perioxiomes, these are a special type of lysosome. These are really only going to be found in an area like the liver and the spleen, where we are filtering and cleaning our blood, so to speak. And so in the liver in particular, these perioxiomes are going to be in the hepatocytes, the liver cells, and as the digested material that is in blood comes through there, it will get rid of things that it needs to get rid of, particularly alcohol and things like hydrogen peroxide, which is a byproduct of some of the metabolism that's going on. And so a perioxiome is simply a specialized lysosome. Now, in this picture, it's kind of showing this is an actual picture of a mitochondria. And what's really important when we get into cellular respiration, the third phase of cellular respiration is called the electron transport chain. And what's going to happen is inside the first little membrane in here, we go through what's called the citric acid cycle. And its byproducts end up being what we call high energy electrons, and they get trapped in between the inner membrane and outer membrane. And as they move through, there's a little turnstile here called ATP synthase. And every time one of these high energy electrons moves through, it produces an ATP. And it's really where we get most of our energy from. And again, that's why the mitochondria is called the powerhouse of the cell. And again, we're going to study that more in the next chapter. This part here is kind of the cytoskeleton. There's three types of fibers: microfilaments, which are rods; microtubules, which are tubes, duh. And then these little, what we call intermediate fibers. And they kind of like the name says, they're not the super small, but they're not large. They're kind of in the middle, and they have several different types of proteins in them. Um, but this is not something I'm going to concentrate on this chapter. I hope you understand that these three make up what we call the cytoskeleton, which is the framework for the cell to give it its shape, and help structures have places to attach or move. I would hope if I was asking anything on these particular, I might ask which one are tube-like, and I would hope you would know that would be the microtubules. All right. So anyway, I'm not going to spend much time on this. This picture kind of shows how they are here as they're, the different areas and whatever, microfilaments, microtubules. Um, but again, not something I'm going to test you on. Getting back to some of the structures that would be test material, the centrosome. Now, the centrosome, which means central body, consists of two centrioles. Right? There are two little things in the centriole. Now, the way that I remember it is in old school, you used to have cities that would have these big walls around it to protect it. And you'd have like a gate in the middle. You'd have these little towers around it. And you'd have a person that stood there, and they would watch out over the countryside to see if anybody was coming to attack. And their name, what their job description was, is they were sentries. They were on the lookout. And I was always told that's where this name comes from. All right, the centrioles are on the lookout because what they're doing is they're watching for the cell getting ready to go into mitosis. And so once that happens, these two centrioles, which again are found in the centrosome, they will divide up. So if this is the nucleus and this is a cell, it doesn't look anything like that. Both of them would be in this little area called the centrosome. When it comes time for the cell to divide and go through mitosis, one will go this way and one will go this way, and they will be the point person to pull the cell from side to side to have it divide. This is kind of showing where they are. Not a big deal on this.